Welcome to Wednesday. Here is our plan for today. We're going to continue the same pattern. We're going to cover the same learning targets, but instead of carbs and lipids, which we have already finished, today we are covering proteins. Buckle up and get ready for quite a lot of notes. Proteins are the most complicated of the four macromolecules. There's a lot of information that we have to learn about these. So get your note sheet out and get ready for some really fun information, but a lot of information on proteins. Proteins is that they are made with almost all of our 6 most common elements in living things. They're made of carbon, hydrogen, oxygen, lots of nitrogen, and sometimes sulfur. The monomer, or the individual building block of every protein, is something called an amino acid. So if you look at the diagram I've got up here, amino acids are these little beads. You string those beads together and it makes a peptide, or a simple protein part. And then you tangle that all together and that makes a protein. Each individual amino acid looks something like this, and you should have that diagram on your note sheet. You're just going to need to fill in a couple of the letters here. But the basic structure of all amino acids is that they're going to have this amino group with a nitrogen on it, and then they're going to have a carboxyl group, or an acid component. That's why they're called amino acids. Aminos and acids. And then in the middle, we've got this thing called an R group. And the letter R is not an element. It's not on the periodic table. R just stands for variable. It means that it can change. There are 21 different varieties of amino acids that naturally occur that we use to make proteins. And you can plug just about anything into that spot, and it's still considered an amino acid. So, for example, the way I like to think of it is that amino acids are kind of like a ring. And you can plug a whole bunch of different things into that ring, and based on that, it has a different purpose. So if you have a ring and you stick a diamond in it, well, that means something. But if you take a ring and you put a giant ring pop candy on it, that means something way different than a diamond ring. But they're still both rings. Just like an amino acid, it's got the same basic structure. We can plug a whole bunch of different things into that spot. Examples of proteins. It's important to see that there are 4 different ways that proteins can fold up, and based on those ways that they fold up, they have different functions or different purposes. So the first level of protein structure is how they're folded up. So that's this one right here. Kind of just looks like beads on a string. And if you can imagine that, if you've ever had like a cord or an actual set of like a necklace, or even just a rope, and it's all loose and it's just one single rope and it's not tightly coiled, it's going to look something like this. And that's the primary structure of protein. That would be a protein that is used for muscle fibers, for example. Secondary structure of protein is either going to fold up into this pleated sheet, looks kind of like a zigzag thing, or it's going to coil up like a little spring, which is a simple helix. Secondary structure is going to cause a little bit more complexity. These are like the proteins that are in our face and in our skin. Collagen would be one of those proteins, and that's what allows your face to spring back after you smile and you don't get wrinkles. But over time, as we know, old people don't have that protein as much, and that's why their face, they smile and it stays deep wrinkles wherever they had smile lines. Tertiary protein is really tangled up. So what I want you to imagine here is you had a rope, and it was a nice simple rope where you had a chain or a necklace or even a cord from your earbuds, and then you started to twist it, and you got this shape. If you keep twisting and twisting and twisting and coiling, eventually it's going to kind of knot up around itself, and that's going to form a 3D structure protein. This will be things like hemoglobin. They are meant to trap and hold oxygen to carry it through your blood. They're going to have a very specific shape so that they can hold on to things. And then quaternary proteins, which are 4th degree proteins, they fold together not only in on themselves, but they fold together on multiple proteins. And that's what's going to make our enzymes. And they have a very unique shape that allows them to do their job. And in order to do that, they have a lot of complexity. The function of proteins, some of these first 3 levels of proteins here, their job is to repair body tissues, so building new muscle fibers with our primary proteins. Building new collagen proteins to hold our skin onto our face. That's important. These guys right here would be used for making pigments. So all of the pigments that allow you to get tan in the summertime or have freckles on your skin, those are all third-degree proteins or tertiary proteins. And then the fourth-degree proteins, their function is to function as enzymes. And I'm sure you've heard of enzymes before. Maybe you've heard of digestive enzymes, but we're going to talk specifically about all enzymes and what they do for biology. Enzyme. In biology, an enzyme is anything that is a highly folded protein, like we mentioned on the previous slide, a fourth degree of fold of proteins, and their shape, their unique shape, allows them to catalyze reactions. I don't know if you've ever heard of a catalyst before, but a catalyst is something that can speed up a reaction without being used up in the reaction. So a catalyst for your car is not the gasoline that you're adding, because yes, the gas helps it go faster, but the gas gets burned up in the process. A catalyst in biology speeds up that process by lowering the activation energy or the startup energy required for that reaction. So the way I like to think about it is that activation energy is like hurdles. You have to jump over those hurdles in order to start your race. Now, if the hurdle is really high, the activation energy is really high, it's going to give you a slower start to your race. You're going to move slower. But if those hurdles are really low, you just step right over them and you get to keep running as fast as you want to. So lowering the activation energy or the startup energy is really helpful. It allows all of our chemical reactions that an enzyme can help us with to happen so much faster. So let's take a look at how that process actually happens. With an enzyme, every enzyme in your body is designed to specifically work on one substrate. So for example, you have an enzyme in your mouth, in your saliva, that is specifically designed to work on starches. So if you hold a raw potato in your mouth, this is a really weird experiment, don't try this at school, but if you hold a raw potato in your mouth long enough, the enzyme that is in your saliva will break down that starch into sugars, individual sugars, and that chunk of raw potato will start to taste as sweet as an apple, which is very strange. But that's one enzyme. You have additional enzymes in your mouth that are designed to work on fats. You have additional enzymes in your mouth and in your gut that are designed to work on proteins. They don't work on separate things. Each one is designed to work on just its one target product. So that target that it's designed to work on is called a substrate. The substrate is the molecule that binds to the enzyme and is going to be transformed by the enzyme. A product is what you get at the end of the reaction. This is what happens as a result. After the molecule and the enzyme are combined, what is released at the end is that product. Shoot, this was not supposed to have worked that way. You're supposed to see the whole animation. Let's see if it starts again. Nope, let me try again. So there's the substrate, the little red S looking thing. The enzyme grabbed onto it and shifted everything around, and now we have products that move away. Now you saw it. Excellent. Totally cool, and they're really important for our bodies, but they are not perfect because enzymes can also be denatured. They're very sensitive to changes in heat and changes in pH. So if you warm up an enzyme too much, it's kind of unraveling that tangled up structure that you had already created. If you change the pH too much, it does the same thing. Once the enzyme changes shape, it cannot fit its substrate, and therefore it cannot do its job properly. So an analogy or an example of that is with Pac-Man. So Pac-Man, imagine he's the enzyme and the pizza is the substrate. He is designed to fit this nice perfect wedge of triangle-shaped pizza, and that's going to go together just fine. Thumbs up. But if you heat up Pac-Man too much, he's going to change his shape. He's going to get all wibbly looking here, just like the actual enzyme would start to change the shape of its openings. Now the pizza can't fit anymore. Pac-Man can't work on it. Thumbs down. This is not a working enzyme. So we would say that that one is denatured. So once again, denaturing is when you change the enzyme's shape and therefore it can no longer function. And two things that can do that are heat and a change in pH. So what about homeostasis? How could these things affect homeostasis? Well, we know that without protein, Our body can't repair tissues or build other macromolecules, and we wouldn't have enzymes to help our metabolism. So maintaining our metabolism, that is the job of enzymes. And this is another fun fact, totally cool, just in case you ever want to check it out. Hemoglobin is part of our blood, and you need it. You make hundreds of millions of molecules of hemoglobin every single day. And to assemble even one hemoglobin molecule would take you 78 million years if you didn't have enzymes. But with enzymes, you're cranking them out every couple milliseconds, which is pretty cool. So here's a quick summary of all of the biomolecules. We can look at their monomers or their building blocks. And we haven't talked about nucleic acids yet, but it's part of this animation, so we might as well talk about those. Those are built of little nucleotides. Carbohydrates are built of little monosaccharide sugars. Lipids are built of glycerols and fatty acids. And proteins, as we mentioned today, are built of amino acids.